some of those, okay? There was a question somewhere? No? Okay. All right, so where were we last time? We were talking about transposition, and I want to give you guys a problem and see if you can solve this one. This was actually an observation. You remember I was talking about, for example, the E. coli chromosome, and we were talking about the GAL region, galactose utilization, and they could score for insertion sequences that might be in the GAL region. They were actually just looking for any kind of mutation in GAL. Okay, when we talked about mutations, what kind of mutations could you have in GAL? Transitions, transversions, frame shift, stop codons, right? All those kind of mutations. But they found another class, which is weird, and that's the insertion sequence class. And when they analyzed them, they found that there were, if we take a look here, in some cases, they found some large element, say maybe about 700 base pair, that was inserted. And later we came to realize these were insertion sequences, which are elements that almost always have inverted repeats on either side, and they code transposase for an enzyme known as transposase. And transposase has the ability, just like a restriction enzyme, to cut DNA. And what it does is it recognizes these inverted repeat regions, and it cuts there, and it can also cut a target DNA staggered like that. And it can cut these out and put them in here. And so that's transposition. That was the jumping gene. That's moving genes. That's how you move. But here's the observation. They had a bunch of GAL mutations. Let's call them GAL1, GAL2. We've got, let's say, 2 mutations that we're looking at. And what they found, surprisingly, is 2 of these mutations, they're called GAL1 and GAL2. When they isolate, now this is the GAL output, right? And now remember, when I say 5' and 3', I have to say 5' and 3' if this is on the chromosome. 5' and 3' relative to what? Right? You guys see what I'm talking about? Because I could take this whole thing and flip it over, and now you'd say, well, it's reversed. But it's 5' to 3' if I look at this top strand relative to, let's say this is the lac operon, and let's say that the lac operon promoter is going in this direction, and the GAL promoter is going in this direction. Then we could say this is a 5', 3' top strand relative to the lac. You see how now I can say you can't flip this on me, right? Because I've got some kind of a landmark that I've got relative 5', 3'. So here's the point. If you take 2 of these mutations, GAL1 and GAL2, and take the 5', 3' top strand. So we got 5', 3'. I'll call it the top strand because the way I drew this is the top strand, right? And we take another 5', 3', one from GAL1, one from GAL2. And we do heteroduplex mapping. We anneal them, and what would you expect? What would you normally expect if you just took 2 5', 3' sequences? Are they going to hybridize? Okay, here's the results they got under EM. What does that say? What does that say about transposition? What does it say about insertion sequences? What does it say about orientation? You should know this right off the bat. Even being Friday, you should know this. Okay. What it says is that this insertion sequence in one of them was in one orientation, and the sequence in the other one was in the other orientation. Remember how I told you to flip DNA a long time ago? And then you took the 2 strands, and you found out that 2 top strands could hybridize? How was that possible? They're not hybridizing 5', 3'. The hybridization isn't occurring like this, is it? No, because DNA is antiparallel. That doesn't work. So what does it mean if I've got hybridization that can occur like this, but it's like this? What does that mean? It means means that those two DNAs were flipped relative to each other. We flipped them. Well, what do I mean flip? I mean the insertion sequence in some cases could go in one orientation when it hops into gal, and it could go in the other. There's no orientation to transposons. They're randomly going to go in one direction or the other. Why is that possible? We're going to talk about that. The mechanism of transposition is such that the transposon doesn't really know which end is which, and it can put it in this, this is a DNA right here. It can put it in like this, or it could have grabbed it and put it in like that. Put it in like this, or flip it, put it in like that. Everyone got that? All right, and so you see the data that told them that insertion sequences could go in either orientation? Hopefully? That makes sense. Okay, so what about transposition? When transposition happens, normally we see a characteristic repeat sequence where they're inserted. So what I mean by that, let me give you an example. Here's an example. This is a transposon insertion sequence 3, IS3. And IS3, so IS3, if we look at IS3, what it looks like, it actually has inverted repeats. It encodes for a transposase, so I can say TPase, transposase, and an additional protein. And these are the inverted repeats. I'll abbreviate them IR. So this is what it actually looks like. And when it actually inserts, if we look at this transposon after it's inserted, we see something like this. It's going to be, and this is just true for this transposon. So where it inserts, we have a little repeat of 3 base pairs. Where it inserts, we have a little repeat of 3 base pairs. And this repeat, how the heck does that happen? It happens as part of the transposition. I'm going to show you that in just a moment. But I want you to just note that there are repeats. And then the inverted repeats would be here. So this element still has inverted repeats right here. These are little IR sequences. Doesn't lose those. When it's inserted, it's got the inverted repeats because that's what transposase is going to recognize. But what we're going to find out is that when it inserts it into a target, we get a repeat, a direct repeat. Sometimes there are 3 bases, they can be 7 bases, they can be 5 bases, they can be different sizes. Usually not too many bases. Not like 50 bases. But a small repeat. Okay, what about, just before we talk about mechanism, which is replicative versus conservative, let's talk a little bit more about what the heck transposons are. I'm going to give you just some transposons here. I just want you to be familiar with how they're built. Let's see. Say IR, 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 IR. This would be something called TN5, transposon 5. We use this a lot in the laboratory. What it is transposons. TN5 is a composite. These guys have just been built by nature, just by, how does it get built? Well, IS50 is an insertion sequence where you can, this guy, IS50, encodes a transposase that can actually transpose and cut this end and move just the IS50. But the transposase enzyme can recognize this inverted repeat and this inverted repeat and carry the whole thing, this whole region, and insert it anywhere, which will give any bacterium that it inserts it into a kanamycin-resistant and streptomycin-resistant phenotype. Okay? So this is called TN5, and TN10 we're going to talk about. TN10 is kind of similar. It has an insertion sequence 10, inverted repeats around it. it carries tetracycline resistance. This is a natural transposon, all these are. And IS-10 here, and that's TN10. So TN10 can transpose using these outside, we call those outside inverted repeats, and tetracycline will go, right? But the other thing is it can just transpose the insertion sequence itself, but it won't carry any resistance. Can you see why these transposons can provide bacteria with a really quick way of gaining resistance? These transposons can hop in the chromosome, and many times they hop into plasmids. And I mentioned a plasmid, and you don't need to know all of this, but this is one that just came out in the literature. I told you about these terrible outbreaks, things like Klebsiella that cause urinary tract infections, and they're killing people now in hospitals. And here's an example of a plasmid that's just isolated. It's about 24,000 base pairs. And what does it have on it? Well, it's got everything we're talking about. So, for example, it's got a transposase, TNPA transposase. It's got something called KPC2, which is a carbapenem hydrolase. This is a big deal. You see, here's the problem. Penicillin was one of the first antibiotics ever discovered, and it has a beta-lactam ring. And penicillin was super effective. It saved a lot of lives. World War II saved a lot of lives, saved a huge number of lives. And the problem is bacteria make penicillinases, and the penicillinases have spread so much that penicillin isn't nearly as effective as it used to be. Carbapenems have a beta-lactam-like structure, but they are resistant to penicillinases, which we can call beta-lactamases. Those are enzymes that cleave the beta-lactam ring and destroy the penicillin. Here's the problem. This is very new. This new KPC2 gene encodes a carbapenemase. And so that thing is spread around because on either side of that, you've got these insertion sequences, basically, IS elements, that can move this thing onto different plasmids. This plasmid also has, it's got amikacin resistance, which is another antibiotic that's used quite a lot. It's got an addiction module like HOX/SOC, so it can't be lost by the host. I'll just say like HOX/SOC. It's got a coli-1 origin and a ROP protein. Remember what ROP did? ROP helped the RNA1, RNA2 hybrid occur so that we could initiate and regulate, right? Replication. Does everyone remember that? Okay. And it's got one more thing that we're going to talk about when we talk about conjugation. It's got what's called an ORI-T, also known as MOB. Very well-known sites. ORI-T or MOB. And we'll talk about those. That's an origin of transfer. We're going to find out. I talked to you about rolling circle replication. That's the initial cut. Is made at ORI-T, also known as a MOB site for mobilization. That allows this plasmid to be mobilized, mated to other bacteria. So it can move by mating. Okay? There's a mating system. We'll talk about how that works. This guy can move from bacterium to bacterium, even from E. coli to Salmonella, easily. So here's a plasmid that just probably arose in the last 10 years. And now it's in the United States. And it's scaring the hell out of the CDC. So these are the kind of things that make what you're learning about incredibly relevant because we're trying to figure out how to solve this problem. This, you know, can you imagine going to the hospital with a urinary tract infection and there's no antibiotics they can use to stop it. And that's where we are in some cases. Okay.
What do they call the plasmid?
This plasmid, what is that plasmid called? I'm not even, let's see, this thing is called S15. Plasmid S15. It's a plasmid that was just isolated and identified. And there's a lot of them. That's not the only one that carries KPC2. KPC2 is the scary thing. That's this metalloenzyme, and it cleaves carbapenems. And carbapenems are the last resort for these particular bacteria. Okay, a couple more things, and then we'll be, I just really want to talk just a tiny bit about how do you introduce a transposon into E. coli, okay? So let's pretend that we have a bacterium here. And let's say there's a plasmid, and we have TN10. And we're going to learn a little bit more about TN10. And you know what TN10 looks like. It carries tetracycline resistance. And this is one bacterium and this is another. And let's say that I can purify this plasmid that has a TN10 that carries TET resistance, TET-R. And I want to introduce it into this bacterium, and I want to get a hop in the chromosome. Why do I want to get a hop in the chromosome? Because where that transposon lands is probably going to disrupt a gene, and I can use this to mutagenize bacteria. Pretty cool. I've got tetracycline resistance linked to it. Here's the thing. If I introduce this plasmid into this cell has a chromosome. The plasmid will come in, and the TN10 will be there, and maybe it's going to hop into the chromosome a few times. But the problem is the plasmid that carries it replicates, right? You assume it probably replicates. And if it replicates, this cell will be TET resistant. You guys agree? Be TET resistant. But am I selecting for hops in the chromosome? No, I'm just selecting for the plasmid to come in, right? How do I select for hops in the chromosome? I want to have the replication of this plasmid conditional. What I mean by that is that one condition, it will replicate, in another, it won't. So we have plasmids that only replicate if there are certain genes that code for proteins that allow it to replicate. So for example, there's a plasmid known as R6K, which is the most common one, that requires a little gene called the PIR gene, PIR, and PIR provides something required to replicate that plasmid. It requires something it requires something that's involved in the initial replication of the plasmid. So as long as this cell has PIR, the product of the PIR gene is something called pi. Pi binds to R6K, lets it replicate. Okay, fine. We get tetracycline resistance in this cell, and everybody's happy. You purify the plasmid and put it into a cell that's PIR minus, that doesn't have the PIR, and when you put the plasmid in, what's going to happen? It can't replicate. But if I select for tetracycline resistance, what do you think I'm going to get? Any TN10 that escaped the plasmid and got to the chromosome will replicate with the chromosome. And the plasmid will not replicate and will be lost by attrition. And if you select for TET resistance, you will have selected for the TN10 to hop into the chromosome. Does everybody see that? Okay. So that's how we introduce transposons into bacteria. We don't have to use a plasmid. We can use a phage that has conditional replication. The phage carried in, and now the phage can't replicate. And basically, think of the TN10 as a little parasite, and it's growing on. It's on the plasmid, replicating with the plasmid. If the plasmid can't replicate, that little parasite is now going to. What's happening here? People are leaving. So, something I said? All right. So the TN10 is like a little parasite, and when it comes into a host and it can't, and the plasmid can't replicate anymore, it can escape, can it? How does it escape? Cleaves itself out of there, moves itself to the chromosome, and the chromosome can replicate, right? Forget the plasmid, right? So that's the idea. And we call these vectors, like the ones that I just talked about, called suicide vectors. A suicide vector is one that when you move it into a cell, an E. coli cell for example, it dies, it commits suicide. It doesn't really commit suicide, it just doesn't replicate. Essentially committing suicide, it can't replicate, and then anything that was on there, like a transposon, if it could hop, we could select for those hops. So, we're going to go back to the question I had. How in the heck do you get those direct repeats? So let's talk about TN10 transposition, which is done via we're going to call it cut and paste, also known as conservative. You're going to see what that means in just a second. So if I have TN10 right here, and I have a target here, what's going to happen is TN10 carries a transposase. And the transposase recognizes the inverted repeats that I talked about. And what it's going to do is it's going to cut, and it's actually, in the case of TN10, it's going to make cut this and have a blunt end, and it's going to cut this in a staggered format. So it's going to look like this. Is everybody like this? Everyone see what I did? I cut here and I cut here just like restriction ends on. And now I'm going to pull these apart. Everyone see that I get this and I get this. Everybody with me? Okay, so this is the target. In the case of TN10, it actually has hot spots, places that it likes to cut more than others, which isn't so good for us as geneticists if we're looking for random mutations, right? If we're looking for random insertion of a transposon, TN10 isn't my favorite one. TN5 is another one we use in the lab that doesn't so much care. It inserts randomly. TN10 actually cares, but what's important is that it makes staggered cuts here and it makes blunt cuts here, and what it does is inserts itself just like this. This gets ligated, and now here's the weird thing. This is so weird to me. This TN10 literally is lost from this location. So when it cuts, this guy here is lost. The reason we call it conservative as opposed to replicative is that we lose it from this site and we gain it in another. It doesn't make a copy of itself. It cuts itself out and it goes to a new location. Conservative. And what do you think happens? It's not really good there. What happens here to these sites? Filled in? What do you think fills that in? DNA pole 1, probably, right? What is that? What did it just do? What's that sequence and that sequence? Didn't we say that the target's somewhere back here? Didn't we say ATT, direct repeat? Now do you see how that happens? How did that happen? Magic? Not magic.
Staggered.
We cut staggered, and when we cut staggered, we leave a region here that's the inverse or complementary to this region, right? So if we fill this in and fill this in, what do we have? A direct repeat. That's why one reason I've been trying to build this idea of 5', 3', and all these little things you do in your head, because you should be able to see this. And if you can't, it's okay. Make up a sequence and then see what happens. Insert it into your favorite. Just make one up and see what happens. When you fill it in, just try it. It's worth it. Okay, so we call that conservative transposition. And this one is a little bit more complicated. So TN10 is an example of conservative cut and paste. Another transposon called TN3, and there's a lot of them. Okay, TN3 is not the only one. TN3 does a replicative transposition. And what I mean by replicative is if we look at TN3 and a target, when we're done, if this is TN3, when we're done, the target's going to have a TN3 and the donor's still going to have a TN3. Okay? We didn't lose it. The donor didn't lose it. It's not cut and paste. We didn't cut and paste. What we're going to do is we're going to do it all with the same kind of idea, but here's the differences. The first thing is we have a staggered cut. And by the way, I'm going to put up a nice picture of this today. I've got a picture I have drawn out of this, so you can try to copy this down. If you can't, don't worry. I have a nice picture. It'll be on GouchoSpace, okay? In color, just like this. So here's what's going to happen. We're going to cut it staggered at the TN3 and staggered at the target. Now remember, how did TN10 work? Blunt end cuts, right? Okay, we got staggered. The transposon is, these cuts are pretty far apart, so to scale, this would be huge compared to the target. Okay, the target is, these are only a few bases apart. This is the whole transposon, but I don't want to make it huge because we're not going to be able to do this. So imagine, this is the whole TN3, these are the outside inverted repeats. Right? The IRs on the outside, just like we've been talking about, nothing different, but we're cutting staggered. Now here's what we're going to do. When I cut this, it's the same idea as the restriction enzyme. I'm going to have this, and I'm going to hook this end up to here, one strand, and this end to the bottom. So this top guy goes to here, this goes to the bottom, and what I'm going to end up doing is having a gap of the entire transposon plus that little region of target. And I'm going to do it again down here because I've only got one strand. So I'm going to have to, these little lines here are what? I've got a gap, single strand gap from here to here, single strand gap from here to here. How do I fix those? I fill them in with DNA pol 1. If I fill them in, what did I just do? Duplicated this whole region, did I not? How did I duplicate it? What I did is I just staggered cut this thing, and I only hooked up this to this and this to this. And so what's happening is essentially I'm using one strand as a template to make the top strand, and another strand to make the bottom. I'm using this twice. I'm using the top here to make a complementary, and the bottom here to make a complementary. Voila, I have two copies. So replicative means replicative. Why did I say replicative? Well, because there's some replication involved. Isn't this DNA replication? It is. It's not huge, but this is probably 800, 900, 1,000 base pairs or more. Does everyone see that? That's all replicative transposition is. And now, here's a problem. Okay, we got a problem though. If we're in the transposon, we got a problem. When we do this, whoa, if that's in the chromosome, if this target is the chromosome, we have one big circle. It's all part of the chromosome, isn't it? It's integrated into the chromosome, right? But that means the donor is now integrated with the target. So the transposon has a way to resolve itself because inside these guys here, now this is going to be a little messy, I'm sorry. And inside here is something called the res site. Do you remember the CER site that some students have asked me about with plasmid chole-1, for example? The SIR site? That's the site of recombination so that the dimers go back to monomers. Remember that? This is the same idea. This guy, this res site, there's a site-specific recombination that will occur between here and here. And what is that going to do? Going to give you these back, correct? But what's going to be different about the target? It's going to have the transposon, and the donor's still going to have the transposon. Does everybody see that? All a res site is a way for it to resolve back to the donor and the recipient. Okay? And why does it want to do that? Well, because it doesn't want to lose a copy. I mean, here's the problem. If it's just all integrated in the chromosome, then in a sense the transposon is lost from the donor because the donor is incorporated into the chromosome, and you've got to get the donor back. If the donor is a plasmid, there'll be a selection for that because that plasmid, like the one I just put up on the board, the nasty plasmid that's out in patients right now, that plasmid, there's a selection for that thing because every time it goes to another E. coli, for example, in a pig farm or wherever it is, it gives it resistance, and so that guy's going to be selected for. That's why when we give antibiotics to animals, for example, in massive doses and people, we're selecting for these nightmares, these chimeras, these nightmares. Chimera means an amalgam of all kinds of things. This guy has got everything in the universe on it. It wasn't magic. We selected for it. And how did that happen? Because transposons are amazing. They can carry pieces of DNA that allow them to transpose. And let me give you just one last example of that. And this is the last concept I want to give you on this. See if I can put this up here. All right, so here's an example of TN10. I'll call this the versatility of transposons. So you can put this down. This is a real straightforward, just a little diagram of, let's say this is a TN10. We've got tetracycline resistance. We have IS10 here. We have IS10 here. That's TN10, isn't it? Right? And let's say it integrated in a plasmid that has ampicillin resistance. That's a plasmid, right? And it's got, let's say, the CoLE1 origin, ORI-V. That's what we call it, right? ORI-V. ORI-V, like that. Okay. And now remember, this IS10 has inverted repeats. Here, 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 here. We're going to call these guys the outside IRs. And we're going to call these guys the inside. Outside. You got outside. Why did I call it outside? Well, because for TN10, if you cut here and here, the whole thing goes. IS10, IS10, tetracycline resistance. Everyone see that? So far, so good? If this TN10 integrated into this CoLE1 plasmid, that's the CoLE1 plasmid we've been talking about all this time, okay? Turns out, you know this, I just told you this, transposition could occur with these two guys, right? Not the outside, but if you use these inside guys, then what's going to happen? You'll transpose all of this, but not the TET. So now we can transpose and move AMP. Does that give you an idea of the power of transposons? If not, let me repeat it. TN10 can hop as TN10. Okay, that's happy old TN10. It moves the whole thing. IS10, IS10, TET resistance, it moves TET. What if there's a selective pressure for AMP resistance? If it hopped into this plasmid, all it's got to do is use these ends and move everything between here and here. So all of that gets moved, carrying the IS10s on the ends. Now it can move AMP, or anything else that's on this plasmid. What if this plasmid has 10 resistances? Then TN10 comes in, and now it can move all 10. How did that happen? It used the inside ends. It has outside ends, inside ends. Pretty darn neat if you want to have some amazing evolution occurring. Okay. Let's see. And there's one more. Didn't really want to spend as much time in transposition, but I got to give you this one last experiment. Okay? Man, I just lied to you. I said I was going to do the last thing I said. Okay. I got one more thing. It's all having to do with TN10. So I'm just going to ask you a question. So a really brilliant scientist, her name is Nancy Fleckner. You don't need to know her name, but she's at Harvard. She's an amazing scientist. She did this experiment, and I think she was the youngest female biology faculty member who got tenure at Harvard. I can't remember. Anyway, she's quite a scientist. She designed this experiment to test whether TN10 was conservatively transposing or replicatively transposing. Now, I told you it was conservative, right? You know that. But she didn't. Nobody else in the world knew. So here's how she set up the experiment, which is pretty cool. She took a lambda, actually two lambda phage. One of them was LAC plus, and the other was LAC minus. The LAC minus had a mutation, say in LAC Z. Okay. So it had a LAC Z mutation. And what she did, this is really cool, these were carried by phage. It's sort of incidental. I mean, she used phage to move this thing back and forth, and you know about phage. Phage lambda, we talked about lambda. And what she did is she denatured this guy, and denatured this guy, and mixed the top strand from here with the bottom strand from here. Pretty weird. And now she's got the lag Z minus bottom and the lag Z plus top. She did this in a test tube, because if she did this in a bacterium, what would happen? Right, the DNA repair system would fix it, right? One way or the other. It would recognize a mismatch, would it not? Yeah, because the lag Z minus mutant has a mutation. Then what she did is, this is all in vitro, all in a test tube. There's no enzymes to go screw this up. It's a beautiful hybrid. And then she packages it in lambda. So she takes this guy, it's all part of lambda, okay? So there's lambda phage, lambda phage. She packages it in phage lambda, puts it in a phage head. Oh, she doesn't put it in. Lambda puts it in, because lambda knows how to package DNA. That's what its thing is. It knows how to package its own DNA. And so what happened is she takes these guys, and she infects E. coli. So here's E. coli. Here's our phage. Here's our DNA. Sorry, yeah. Like that. It injects it. Now, the key is, this LAC-Z gene is all within TN10. So within those ends of TN10, the ones we showed, she put a LAC-Z gene in between. You got that? So when TN10 transposes, the LAC goes with it, right? So I'll say TN10. She injects it. Now this phage has AMBER mutations in the head and tail genes, and this strain is no suppressors. So I'll call it sub-0, and we have 2 AMBERs in the head and tail genes. You guys understand this now. I can grow this phage in a suppressor strain that has a tRNA that suppresses the AMBER mutations. You know all about that. I don't have to tell you again, right? You know all about this stuff. So I can, sophisticated genetics, but you know how to do it now, right? Sub-0 is a strain that has no suppressors. Therefore, that heads and tails won't get made, and what happens to the phage lambda? Once it injects the DNA, it can't replicate, so what are we using it for? Just to do this, to inject that, TN10. Remember how I talked about the conditional plasmid, and you can introduce a transposon with a conditional plasmid? This is a conditional phage. Isn't an AMBER mutation with a tRNA suppressor conditional? If you have a UAG stop codon, it normally will block translation, right? But if I have a suppressor tRNA, what can it do? Suppress it and allow it to be made. Isn't that conditional? Yeah. So now here's the thing. This is the cool part. So what she did is she then injected this, and now the only way this guy is going to, the TN10 is going to survive, is to hop into the chromosome. Hop. And when it hops, if it's a cut and paste, it'll look like that. You agree? It cut and it pasted. Right? But if it's replicative, is it going to look like this? No. If it's replicative, this guy's either going to be like that, or it's going to be like that. And why is that the case? Because remember, when we looked at replicative, we're using one of the strands. If it was blue, we're going to make a blue. If it's red, we're going to make a red. We only have one strand here, so we're not going to have an amalgam of 2 different, like a cut and paste will give you this. Could that ever occur with a replicative strategy? Look, it can't, because we already have one, we're just making a copy of it. So of course they're both going to be the same, because we're using this one as a template to make this one. So here's what she did. You want to know simplicity? She took these guys after infection and put them on lac indicator media, which you know about. I think she used Exgal. I talked about that. It makes a blue color when it's cleaved by beta-galactosidase. And here's what she found. Oh, let's see. We're going to make blue and white, just like we did. Blue is lac, I hope I said blue is, I said blue is lac minus. Blue or lac plus. Let's make blue lac plus, okay? So it was a colony that didn't know what it wanted. It had half of it was lac minus, and half of it was lac plus. Literally, the colonies look like that, many of them. How did that happen? If you start like this, then when this cell replicates, what's going to happen? This is used as the template for one daughter. This is used as the template for the other daughter. One daughter will be blue. One daughter will be white. Does everyone get that? But with replicative, will we get those? No, because we will either see blue or we will see white. We will never see a colony like this. Why? Just went through it. Because replicative transposition, one strand is used as a template to make the other. You'll never have cut and paste where you've got a blue and a white together on the chromosome. Blue is used to make blue, white's used to make white. Is that? Replicative uses one strand as a template, so you're never going to have one strand of one type and one of another. They're both going to be the same because you're using it as a template. Okay. I think that's all I'm going to say about transposition, even though I would love to say more about transposition. I like transposition, but we don't have time to talk about anything else in transposition. And what was that? I'm an engineer. An engineer?
Yeah.
Ah. Engineer humor, maybe. I don't know.
They kept spelling it wrong and then crossing it out.
It was there since like week 1.
Okay. Now, in 5 minutes left, I'm going to just give you the very intro to conjugation. So we're just talking a tiny bit about conjugation. So now we're going to move to conjugation. Now, conjugation, I say, is a separate topic, but as you know, I just told you the plasmid that carries this carbapenem resistance, that guy has a conjugation system, it has transposons, so all these guys, all these things are happening all in the same plasmid. So, it's a separate concept, but it's all related. So here's what we're going to say. We're going to focus on the F plasmid. And F is called fertility factor because it's sort of mating. It's sort of like mating. Not really. Here's why it's not like mating. You have a bacterium, and it's going to make these pili encoded by the F plasmid. So the F plasmid encodes pili. The pili actually bind to and allow a conjugation bridge to form in the female, and I use the word female loosely here, and I use the word male loosely, because they really don't bear any resemblance to male and female that you and I are used to. Okay? But bacterial geneticists, they get lonely, they're in their laboratories, and fertility factor. I mean, come on. They're anthropomorphizing beyond belief. But here's why it's not mating. This F plasmid, if it contacts an F minus cell, will introduce itself into that cell, and that cell will now be a male. Imagine how weird that would be in the mammalian world. Wow, a female turned into a male. Everybody's a male after a while. That probably wouldn't work. So here's what happens. This plasmid is going to do rolling circle replication. How does that work? We talked about it. We get a cleavage, a 5' end, a 3' end, and the 5' end is going to be introduced into this female. And what's going to happen is we're going to get discontinuous synthesis, and when we get a whole plasmid worth, there's enzyme that recognizes that and recircularizes and ligates it, and we transfer the F by rolling circle. Now, of course, this F is losing one strand, so how does it make its other strand? can you see that arrow? It's going to be going, and this guy is rolling in this direction, and basically this strand, oh man, that is ugly. So this strand goes there, and now we synthesize. I'm going to peel this away. Can everyone see that? No. That's a mess. Anyway, if we peel this strand away, we can then start synthesizing a new one here, right? Because this strand imagine this guy eventually is going to come over into here, right? That's pretty ugly. Last thing. So if we have, what we're going to do is just synthesize, and synthesize, and synthesize, and this thing's going to roll, okay? Does that make sense? All right. And that's about it. So if you need midterm 2, come up right now. I'll get you midterm 2.